and start the recording. Okay. We have lecture test number 1 next Thursday, the 24th. It's at 2 o'clock. It will be on Canvas, just like the previous test you took, although that was a practical. Okay. You go to our class website, go from there to the very last thing on the page and under modules, and it will be lecture test 1. Okay. And then you just take the test. I think I'm going to probably give 70 minutes, probably make that the norm for us. It was fine last time. I think it will be this time too. And you may take the test at home where you're used to, where you're comfortable. You can take it at the library. You may take it in the parking lot, in your car, with a drink holder. Okay, you can, I don't care where you take it. Okay, I'll get it, you'll submit it. But where you take it may make a difference with the next sentence coming up, and that is we do have lab, our first together lab in the classroom. And the classroom, the lab number is 221. Don't worry, it'll be in the email for you in the science building. And we will start that at 4 o'clock. Now, trust me, in the future, after we get past this first day, we will probably run lecture followed immediately by lab. Okay, so there's a gap between the end of your test of 70 minutes and 4 o'clock. During that gap, you either drive into school, depending upon how far away you live or how much traffic you expect, or you're already here and you walk over from the library, or you're down in your car, or you're sitting on a bench someplace outside if it's not too hot or cold, and you can walk over to the laboratory. Okay. The laboratory will be open. However, when you get there, don't just walk in and grab a seat. I'll have it figured out from the few classes that were meeting actually on campus, how they managed the seating arrangement. Okay. And then we'll have our first lab starting at 4. The portion of that lab that I take up is roughly an hour, sometimes an hour and 15 minutes. I will either be showing models of human musculature, or I'll do a cat dissection. We have cats enough for every table or group to have their own, and that's usually the way it's done. But you need to see the dissection first so that you can follow it. And so what I do, I have a camera, and I do the dissection sitting almost alone. And the camera projects from the ceiling onto a big screen in the front, and everybody can see how to do the dissection. And then hopefully you'll, amongst yourself, decide who is going to do the dissection that day. You can rotate it or you cannot. It's up to you. And then you label your muscles. I know I told you this. After you finish labeling, you call me over. I check to see that they're correct, and they're not always correct. And so they need some help. And I move the labeling pins around for you. And then once you've got all the cats labeled, and once I've got all 7 tables worth of cats labeled, a lot of redundancy, huh? Well, then you can go around and pick the best labeling that you like. Take your camera and take your pictures home with you, okay? Now, you can elect to stay and study in the class because it's going to be a practicum that is done in the classroom using real material, not photographs. But you can always take those photographs home as well. The length of time that you spend in the lab after I finish my talk and presentation. Depends on how much lab time you feel you need. Some people don't feel they need any. Other people will stay right until the end. The end is 6:50, 6:50, 10 minutes of 7. Well, we have never done that online because we've never had the opportunity. But now you have an opportunity to stay if you wish and study in the classroom. If you're going to instead go home and think that you're going to study, as you probably have been, that may actually take more time. If you, on the other hand, feel that no, that you can finish this off at home, or you've got some other obligation that's not my concern, I do not watch the clock. Okay. If you leave early, I don't care yet. If you leave early, I won't say anything yet. If you leave early and you screw up the examination with a poor grade, then I'm going to say a lot. Okay. So when you leave, I don't care. I want you to arrange your time and your study to get the best grade that you can out of the class. Okay. Okay, so on the first day, you must wear a mask. I have to too. I got some conflicting results. Rachel, you put your mask on, you're there by yourself, or where are you?
I'm at the airport.
Oh my God, which one?
Long Beach,
Where are you going?
Sacramento.
Two of my favorite towns.
Really?
No, not at all. I've been to both of those. I went up to Davis near Sacramento because they wanted me
Yeah.
to go to graduate school.
Oh, okay. Yeah. I'm going to from Sacramento to Lake Tahoe. So you have to
Oh,
fly into Sacramento.
well, Tahoe's better than Davis, I'll tell you.
Yeah.
Walked around that campus trying to get a feel for it. And here's a girl chasing a pig. I'm serious, in mud, chasing a pig, trying to bring it down. You know, you have a lot of agriculture there. Okay. I just wondered why you put the mask up, you know. But Haley, where are you?
I'm at the library,
And you
Mission
feel,
Viejo Library.
oh yeah, over there on La Paz and Marguerite. Yeah.
Yeah, I don't know if it's required, but I just put one on anyway.
Oh, I intend to continue to wear them, but it's Saddleback and us. You need to wear it indoors all the time. Now I got two messages, conflicting, of course. One says, "You students do not eat or drink. Do not take the mask off." And the same one says that instructors can turn their back and sneak a sip of water. Well, if you're going to talk for very long, you need that sip of water. I know I do. You see me grabbing water. Today, I'm having Diet Coke. And but then I got another one that's conflicting, as I said. And it said, everybody wears a mask. Okay. Uh, students may lower their mask to sip a beverage. First off, I can't see why you would lower the mask. You might raise the mask and just uncover your mouth, not your nose. Keep that nose covered, please. Okay, I'll feel more comfortable. And sip a beverage. What do they mean by the word beverage? The only thing allowed in the classroom is water, no Diet Coke. Okay, so I'll have to have water too. And if you have a thermos container, you know what I'm talking about, tumbler, that can stick a straw into and it has water or whatever. Of course, I won't know what you're drinking if it's a colored straw, but I'm not going to police that anything actually. Then you can put it under your mask and take a drink. Okay, leave it on the table otherwise. Okay. You can go outside of the classroom, but we have an enclosed hallway. So you have to have your mask on in there too. Everybody uses that enclosed hallway. And so it's a good practice to do that. We'll be right across though from the fire escape stairwell, the outside stairwell. They don't call it fire escape, but I do. And you can easily get outside in an instant, you know. Okay, parking lots. I got a notice, and I think they sent it to you too, that 8, 9, and 5 are some parking lots are being reconstructed. About time, but those won't affect you. Those aren't the ones that you would use to get near our building. Parking lot 5, 5A, and then on the other side, parking lot 7. Okay, but you'll find your way around. Okay. Now, I'm thinking to myself just for a second that I forget something important. We provide gloves for your hands. They're nylon gloves. They do not have latex. So if you're dissecting, you'll always have access to gloves and tools. Don't bring any of that stuff in. You don't need to. Okay, got a question
What if
for
I
you?
have my own dissecting tools? Can I bring those or?
What's going to happen with your own dissecting tools is that they'll end up getting mixed up with our dissecting tools. If you can tell the difference, that's fine. I know you just buy them and you're anxious to use them on something.
Well, I got them in high school in my medical program, so I'm like, I might as well use them.
Well, you're not going to need much. I'll provide scissors, scalpel, but I really tell people the scalpel is not a dissection instrument. It is a destruction instrument. You cut something with a scalpel, you can't put it back together again. So I rarely let people use a scalpel, just occasionally. And a maul probe. We got lots of maul probes. Those are the only things that you really need. So if you want to bring those in, scissors, maul probe, and a scalpel, you can. We used to require all students to buy a dissection kit. Didn't make any sense to me. So I talked them out of it 20, 30 years ago. And now we provide the material. Okay. If you have a couple of extra masks, stick in your pocket or something, maybe in a baggie, that's up to you. Just in case there's somebody doesn't have one and I don't have an extra, I should have extras on the first day, but please don't count on them. They only give me a few, and I'll share if we need to. Okay, anything else? The test on Thursday, strictly lecture test, all multiple choice, 70 minutes. Most of the questions are nothing but words. There are some diagrams and pictures. What pictures? How about the picture of the myofibril? Okay, that's all in the lecture notes. So it's all there. And if you know those lecture notes from the start, way back to day one, when we talked about sutures, syndesmoses, and such, you'll be doing well. Okay. Now, get over here and get chapter 10. Okay. In the book, I'm using edition 7, an old edition. Down on the floor over there, I've got 8, 9, and it doesn't make much difference. I'm going to turn the light on here for a second. Okay. Chapter 10 in my book starts on page 242. It'll be somewhere near there that yours starts with overview of muscle tissue. And that definitely has to be read. Properties of muscles, read it. In other words, just put a check mark in front of it. Contractibility, excitability, extensibility, elasticity. Okay. Terminology specific to muscles, yes. Function of muscle tissues, yes. Types of muscle tissues, absolutely. That's our skeletal, cardiac, and smooth muscle. So page 1 needs to be outlined. There's a table 10.1 that needs to be read. You don't need to outline it. It is all outlined in my notes and in the book. But you need to take a look at table 10.1 and feel that you're familiar with it. Maybe you won't be at the start of the chapter, but by the time you get to the end of the chapter, flip back to it and see if it's gotten any better. Turning the page, mine goes down to check your understanding, which you do not need to do. Okay. And then starts up skeletal muscle. Okay. The skeletal muscle section goes along with figure 10.1, the one that looks like a large piece of beef leg that's been cut, but a piece is pulled out, another piece is pulled out. First, you have the belly of the muscle, the biggest part. Then you have the fasciculus pulled out. Then you have the muscle fiber pulled out. You should be familiar with that drawing. And for the reading, skeletal muscle gets at least 3 checks. The first thing they talk about are the layers: epimysium, paramysium, endomysium, absolutely. The word tendon, a little farther down the page, yes. And then the words origin and insertion are described for you in figure 10.3. That's necessary. Figure 10.2 you can omit. And the portion that follows, nerves and blood vessels, you can omit. So that's a no. So we've only omitted 2 little paragraphs. Muscle attachments, however, we got to pick it up again because that's going to be our origins and insertions. So yes, check muscle attachments. And I'll go on to the next page. Yes. And go down and through, check your understanding. Do the later editions, who has a edition 8 or 9? You're not all using 7, I don't think, are
I
you?
have 8.
I have 9.
Okay.
I have
9.
10.
Do they use the term check your understanding?
Yeah.
Yeah, check
Yeah.
your understanding.
Okay. Well, if that's the case, we're on the same page.
Mhm.
Stop your reading there. Now this time, microscopic and functional anatomy has to be read. The skeletal muscle fiber needs to be read. We've already gone over this. We've already talked about it. So it should be like a review, I hope. Myofibrils and sarcomeres, definitely you need to read that section, the entire section. And please match it up with figure 10.4, which is important. I think you recognize figure 10.4. If you come, it's got 5 rows of pictures. The third row has the columnar looking myofibril, the Z disc, H zone, Z disc, I band, A band, right in the middle, the one that I showed you in our lecture. That image gets pasted into our lecture exam next week several times. Go to the next page. Go down to tighten and stop, not stop, but not include tighten and other myofibril proteins. You might want to read that. Tighten's kind of interesting, but it's we don't need that.
Oh my God.
Okay. Sarcoplasmic reticulum and T tubules. Please read that. I did not lecture on that, but I said 2% of a test can come from the book that was never lectured on. Well, here's the 2%. Sarcoplasmic reticulum and T tubules. You come to check your understanding. And at that point, even though you see quite a few pages left, I want you to stop your outlining and check your understanding. You know you've got the right check your understanding because just below it is mechanism of contraction. The mechanism of contraction is not it's called a theory, but I have the feeling it's a changeable theory. A hypothesis is anything. You say, I think this is caused by that. That's a hypothesis. That's a guess. When it turns out it looks like it's true and it's repeatable, keeps happening, then we up it to the category of a theory. And if the theory holds true, we up it again to the category of a law. But the sliding filament theory, which is basically for the movement of the thin, thick filaments against each other and the contraction of muscle and skeletal muscle at that, the sliding filament theory has lasted for a long time, but I'm sure it's going to undergo some modifications. I just feel it is. And you can go with what we said in the book. I'm sorry, in the outline. I'm getting it wrong twice in a row here. In our notes, you can go by our notes for that. And you do not need to outline the rest of the chapter. Okay. I'm going to take us to our screen. Okay, week 5 assignment. Week 5 assignment is for you to read chapter 10. Now you can always find the assignments in my webpage. I bookmark it myself as Bio 11, but you can bookmark it as you wish. If you go to my webpage and you go to reading assignments, it now includes chapter 10, and that is due at P2. I don't know, to be honest with you, when P2 is myself. I know when, oh yes, I do. It's on the 17th of March. Okay. So you can do that. You can also look at your scores up here. You lost the email that I sent you. You don't know what your score it was. You probably will have that happen to you once or twice during the semester. Probably not on the first test, but if you click scores, then your scores will appear, the ones I sent out to you. Now, in this case, you'll notice that the numbers are all jumbled. Well, so if you know your number, you can find it. I mix the numbers up, or I put them in order as suits me to make the chart up for you. Okay. Back. We talked about the start of the digestive system the other day. I posted a recording for that. We talked about how muscles are named. When we get to lab today, we'll do these muscles. Okay. And here's where we are, the digestive system, part with 2 R's, or part 2. The first part we did, just knowing where we left off, and it was right here. We ended with the stomach and the greater curvature and the lesser curvature. Each have membranes. They're called serous membranes, a term you get on next week. Oh, by the way, there's no class next Tuesday. Okay, no class on Tuesday. Contractual day, whatever that means. Okay, once the food has left the stomach, it exits through the pyloric sphincter. I mentioned to you that the pyloric sphincter is the smallest passageway in the GI tract, and anything that can get through the pyloric sphincter right here can get through anything else. Okay. So the first part is of the small intestine is the duodenum. It curves back under, makes one more curve starting down. There are 3 parts to the small intestine: the duodenum, and then when it starts downward, the jejunum, and later on you'll see when it at its distal end is the ileum. Okay. You'll notice that in the small intestine it's ribbed. We knew the stomach was. The stomach was sort of chaotic in its ribbing, going like more like a net. But the plicae circularis are more like rings that go around the stomach, small intestine. This helps to move the food downward. Helps to move the food downward. So does the fact that more food is coming in, pushing everything downward. Notice for a second a small little opening into the duodenum right here. That's going to get a name. It's a sphincter muscle. It's a sphincter of Oddi, O-D-D-I. It'll be labeled for you in just a moment, okay? And you can barely see in here. Is this green?
Yes.
Okay, the green tube is the common bile duct coming from the liver and the gallbladder. Okay, it brings material in, it deposits it and ejects it through the sphincter of Oddi into the duodenum for the emulsification of fat. Okay. The main function of the small intestine, even though we're only looking at one part right now, all parts serve as the main site of digestion and absorption. Digestion of carbohydrates, much more important here than in the mouth with salivary amylase. We have pancreatic amylase. Here's the pancreas being produced all the time and coming into tubules too and combining right here. You can't see the combination underneath this, but they do combine and they exit together. So we get pancreatic amylase for the digestion of starch. Proteins are also digested in the small intestine. Proteins are also digestible in the stomach. Okay. But in the small intestine, it does a much better job. The third category, lipids or fats, are emulsified in the small intestine. Well, we mentioned the liver, and it's an accessory organ of the digestive system. And this was, an amazing fact to me. The liver has over 500 known beneficial functions. Boy, no wonder liver transplants are so vital to some people. And keeping the liver healthy is very desirable, of course. Here are some of the most relevant, though, functions of the liver. 500, and I'm listing 5. The production of bile salts, sometimes just called bile, and that is delivered to the small intestine and aids in the emulsification of fat. Something's got to break the fat down. The fat really can't be digested chemically. It can only mechanically be broken down to smaller and smaller parts, and the bile salts help to do that. The liver also removes poisons from the blood. We all eat poisons. I'm guessing anyway. Potatoes are slightly poisonous. Oh, we don't feel any ill effects from potatoes, most of us anyway. But if you have a green potato that's been sitting in the sun before you baked it or anything, you've got a high concentration of a chemical called solanine. And the potato is from the nightshade family. And the solanine can really make you sick. Never eat a green potato. Okay. When I was a kid, I would go around the neighborhood. I had secret spots that I knew of and look for sour grass. You know what sour grass is?
Yeah, the one that like you can chew on. It's a bit sour. It has like the little yellow flower on it.
That's it.
And they say like bunnies pee on it, I think, and a lot of kids would like eat it, right?
Yeah, a lot of kids used to eat it. I mean, they do, but not if they know better. It's oxalis is the name of the plant, and I used to, as I was saying, go and try to find a little bunch of it or something. And the kids, they loved it, and they, in order to have some, find out where it was, I made them give me a penny. I upped that to 2 pennies later on. Finally, I got enough to get a candy bar, which was better for me than eating the oxalis, which I love too. The calcium oxalate that is in the oxalis stems will stay in your liver your entire life. It does not go away. So it can lead to some complications like stones, kidney stones, especially. So things that are taken up through the digestive system get into the small intestine and they're absorbed, but instead of going into the circulatory system, first they go to the hepatic portal system, which takes the newly acquired nutrients in the blood, takes it to the liver. The liver gets a chance to detoxify it, break it down, cause it to be less harmful, if it's harmful at all anyway. And then after passing through the liver, it's allowed to get into your normal circulatory system. Red blood cells have a limited lifespan. No nucleus in a red blood cell, and about a run of 120 days. And that's it. After that, they break apart, they hemolyze or undergo hemolysis, and all of this stuff has got to be picked up by phagocytosis and destroyed and absorbed. And that's done by the liver. It's also done in part by the spleen, but the liver's the main organ. I didn't say it at first, but maybe I should have. The liver is the largest gland in the body. Okay. Now, the fourth thing that the liver can do for us is to store excess sugar. It stores it as glycogen. What's the difference between sugar and glycogen? It's like the difference between starch in a potato and sugar after you've eaten a potato. Breaks down to sugar. If you have a lot of sugar in your blood, more than your body needs, okay, it can be stored as glycogen, especially overnight. Okay. Glycogen is an inert form of sugar. Doesn't do anything, it just waits around to be reactivated, returned to sugar when you need it, when your blood sugar gets low. When your blood sugar gets high, the formation of glycogen can proceed. Okay. Well, when you have problems with diabetes, you have problems controlling whether or not the sugar is in the blood or in the tissues. We'll get into that later. But at night, extra sugar is usually stored as glycogen. When you get up in the morning, do any of you drink coffee?
Yeah, like 2 in the mornings when I wake
Yeah.
up.